Hello, my name is Laura Angadi. I'm an independent researcher studying the history of mead, specifically how it was made and what it might have tasted like in historic times. My background includes chemical engineering and biology, which has led me to the topic for today's talk, which is the nature of historic fermentation cultures and how they can be modeled in modern recreations. Now, this might focus on alcoholic drinks, but it's equally applicable to bread and also to other fermenting foods. I'm going to look at three different factors here. First, the insights we can glean from modern scientific studies. Second, the results from the recreation of an exemplar mead recipe using a number of different ferments. And these lead to a framework for selection of a fermenting culture when we're doing a recreation. Keeping in mind that ferment is one of many factors determining the character and flavor of a drink, we know that the byproducts and co-products of fermentation, as well as the materials introduced with the ferment, have a critical influence, which is multiplied when there are multiple organisms in a mixed culture. As well, we want to understand how many thousands of generations of genetic mutation and adaptation may have changed both the function of ferment and the character of the drink produced. In alcoholic fermentation, organisms produce energy from sugar to support their metabolism and reproduction. Our interest focuses on the waste products, CO2 for carbonation and bread leavening, and alcohol to drink. Saccharomyces cerevisiae is the best known of thousands of organisms that use this alcoholic fermentation pathway. But other fermentation pathways are used by other organisms in some ferment cultures. Lactic acid is produced by Lactobacillus, acetic acid by Acetobacter, and buttery tasting diacetyls by Pediococcus. This table shows some specific text references to fermenting cultures. These include ferment, leaven, yeast, barm and lees, which are the top and the bottom fractions of a fermented liquid, sour leaven or sourdough. There is a distinction between bread and ale yeast. Also, we see the use of a wine barrel and the residues in it to support fermentation. The next 5 slides are going to look at different pieces of what we can learn about historical ferment through looking at modern science. The first study gives insight into the breadth of historical fermenting organisms. Yeast were extracted from 4th millennia to 4th century BCE vessels that were presumed to have contained fermented drinks. That these yeast were domesticated for alcohol production was confirmed by fermentation kinetics matching modern brewing yeast, but differing from yeasts on other excavated items. Of the 5 strains that were used to make beer, 4 produced drinks that were good or drinkable to the modern drinker, yet only one of these 4 drinks was from Saccharomyces cerevisiae. In 1996, the first completely sequenced genome, 12 million base pairs, was that of Saccharomyces cerevisiae. Today, over 1,300 yeasts have been sequenced. In 2016, the diagram shown here of yeast strain groups was developed as a simplification of an involved relationship chart. This study found that beer yeasts were more diverse and evolved faster than wine yeasts, probably due to their year-round use compared to once a year for wine yeasts. But wine yeasts were found to perform better under stress of intermittent use. It is worth noting that many milestones of yeast genetics are relatively recent. Lager yeasts emerged in the 15th century, and wine yeasts became distinct from ale and beer strains in the 17th century. Other fermenting organisms also have diverse and complex genetics. Diversity and complexity are a hallmark of sourdough cultures. This study, looking at 500 cultures collected from 4 continents, shows that there are a median of 3 lactic acid or acetic acid bacteria strains and 1 yeast strain per sample of sourdough. Among the yeasts shown here in the lower bar, we see that Saccharomyces cerevisiae is by far the dominant brown here culture. The bacteria are far more diverse. However, Lactobacillus are most common, followed by Acetobacter and Pediococcus. Kveik yeasts and Nordic farmhouse brewing traditions have recently become very popular in modern beer brewing. Again, my focus is on the variety seen in the fermentation cultures collected and cataloged by Lars Garschel, who has been researching these traditions. The Farmhouse Yeast Registry details that over 90% of yeast strains, where identified, are Saccharomyces cerevisiae, with an average of 4 yeast strains per culture. In addition, about half of the cultures contain at least one bacteria. One of these cultures produced the beer Garschel calls world class, and was found to contain 8 Saccharomyces cerevisiae strains plus 1 bacteria. The deposit of this culture into the National Collection of Yeast Cultures contains 8 yeast strains but does not appear to have the bacteria. One of these strains, out of the 8, was commercialized as Omega Lutra Kveik, but 1 strain only. The other 7 have vanished. Finally, I wanted to see what experimental archaeologists are using when they recreate drinks. A review of various papers and presentations showed that a Stringle-strained commercial yeast is the most common ferment, either beer or wine strains. Others have used wild yeasts from open fermentation or collected from fruit skins. A few have used lees from a previous batch, presumably itself using a single-strain commercial yeast. In one case, several yeast strains were combined, and in one case, a yeast loaf was made from a sourdough starter. What can we learn from these modern studies? Historical ferment consistently would have included multiple strains, species, and/or genera, bacteria, and yeast. A pure Saccharomyces cerevisiae ferment isn't implausible, but almost certainly wasn't common. Other materials were present in the ferment, whether as a substrate or a carry-along, and those would have influenced any drink. The genetic links between these historical and modern organisms are understood, but we can't quantify them readily for how they would have affected taste and fermentation kinetics. Wine yeasts may be better historical models than beer yeasts. We do know that the core function of fermentation is the same, as are the rules of chemistry and biology governing organisms. I believe that we can state that modern microorganisms vary within a general range that is analogous to the historical ones. Moving to my recreation, I chose a recipe for originally a medicinal hydromel or mead that is made from 1 part of honey to 8 parts of water. This recipe shows up on the European stage in the 11th to 12th century with translations of Arabic medical writings. It's seen quite commonly and varies in its proportions, how much of it's boiled away, what other additives are made. But it's a good and common exemplar to use for this recipe. The hydromel recipe gave me a must with a specific gravity of 1.074, about halfway between beer and wine. It was split into 8 containers, each of which was given a different yeast. 4 of these were based on the Omega Lutra Kveik yeast, a single strain, just that yeast, 1 with a lactobacillus added to mimic a sourdough, then 2 more that were from a ale ferment that was started 3 days before using the Lutra Kveik, 1 of the barm, the top portion, 1 of the lees, the bottom portion. Then 3 sourdough samples, 1 fed on wheat flour, 1 fed on oat flour, and 1 fed on rye flour, to see the difference that the substrate might give to the taste. The final was left open for a wild ferment, which, given the indoor nature of my semi-urban house in the middle of winter, wasn't necessarily expected to yield significant results. Here we can see the 8 meads all laid out as fermentation was starting and after the ferment was added. You can see on the right that the sourdough options show quite a bit of opacity as the substrate flour mixes into the must. The rye, number 7, was the most vigorous of the sourdoughs, and we'll see that carried out as we go on. The differences that the substrate makes can be seen in this picture of the 3 sourdoughs right before they were added to the mead. The wheat flour was watery and active, and that's the substrate that this sourdough starter has typically been fed on. The oat was less active and quite thick. The rye flour was the most active of the 3, and again quite thick. Each mead was measured and sampled on days 3, 6, and 9. On day 9, all were still fermenting, and you can note that each of them has developed a layer of lees in the bottom, more where there were things present in the added ferment, as in the lees from the ale fermentation and the sourdoughs, and less in the ones where a purer culture was added. The progress of the fermentation over the 9 days for the 8 options can be seen here summarized in this graph using the maximum alcohol if all the sugars were consumed and turned to alcohol as a measure. The ale lees produced from the lees of the barley malt fermentation went almost to completion. This isn't surprising because it would be expected that those lees would not only contain an active culture, but also would contain nutrients in the form of the dead yeast bodies and settled material. The open or wild fermentation was not very successful, and everything else was in between. It's going to be expected that fermentation in most of these would continue over a period of time. The results in terms of flavor are relatively unsurprising. The Pure Kveik was the cleanest of the ferment. It showed some fruity notes, but all those same notes were tasted in all of the ferments that use the Kveik yeast. With the lactobacillus, the perception or the amount of acid grew over time. There's a sharpness from the acid and a slight metallic character that could be stress or could be another component. All 3 of the sourdoughs had relatively little flour character, although the differences between the 3 certainly reflected the flavor of the substrate. The oat had a rounded flavor, the rye was sharper, and the wheat had a somewhat slightly bready flavor. The ale lees had the most side and somewhat off flavors, which again is unsurprising given the nature of the lees consisting of dead yeast and other materials. Depending on the situations, any of these can be considered the suitable ferment for an experiment for experimental archaeology. I've classified each of these choices in terms of the reproducibility over time if you're doing a series of experiments that take several years, interference into flavors, if you're trying to detect details of different malting effects using a yeast that adds a strong flavor is going to interfere with your ability to look at those. Drinkability. If you're trying to recreate a drink for a feast or a public event, your tolerance for off flavors is going to be lower. And then historical accuracy in terms of what we may believe was most commonly used. In conclusion, in terms of experimental design, we can't actually say with certainty that one or another approach is better. We have a large range of organisms and ways of mixing organisms that are suitable for recreation and more or less accurate under certain circumstances, but we need to select a ferment that will optimize our opportunities for data gathering in any specific case, will provide the reproducibility required, and maximize the chances for success given the goals associated with any particular experiment. Thank you all for your time and attention, and I hope I've given you some thoughts that will inspire and help your next drink recreation.